great pleasure to introduce our speaker, here. I guess you already know him, right? He's here and he's very well known for a number of things. And Yuan is a professor at the Department of Science and Mathematics at Weizmann Institute of Science. And right there, he has a very kind of multidisciplinary research team in the area of computational and system biology. And his team has been doing a lot of contributions across several different fields. And during the COVID pandemic, Professor Segal is a senior advisor to the Department of Israel, and he also got a lot of awards. So to save the time, please.
Thank you very much. Thank you for the opportunity to present to you today. And what I want to tell you about today is a large-scale project that my lab has been working on for the past 5 years. We call this the Human Phenotype Project, and part of what I want to achieve in this talk is to tell you about this very massive data set in the healthcare space that we've been building, which actually will be available soon for access, and hopefully gauge your interest in also working on this data set, which, as you'll see, is very diverse, very heterogeneous, and I think has a lot of opportunities for various AI applications. I would say as a computer scientist, I believe that like Benjamin Franklin, investment in data is really, in the end, this is what we learn from, and I think we can see what we've learned, for example, from the Human Genome Project, which brought us the first draft of the human genome in the year 2000. And if you go back to that year, back then we knew only a few variants for some human diseases. 20-something years later, after the publication of the human genome, we now know hundreds and sometimes even thousands of genetic variants that are involved in virtually every human disease. So this really was a major breakthrough in medicine, and already drugs are being developed based on this data. And it's hard to imagine now somebody, for example, with cancer not sequencing their tumor to understand what genetic mutations exist there. So we learned a lot from this, but of course, genetic data is just the genetics we're born with. It doesn't change with life, so it doesn't take into account environmental factors and our lifestyle and nutrition and exercise. And so for that reason, we and others have started to profile additional layers of data that we have among us, and these are collectively called multi-omics data sets. They can be sequencing of our genes, the RNA sequencing of looking at the set of bacteria, the microbiome, looking at the immune system, looking at metabolites. But the way these have been studied typically is one data set at a time in a very kind of narrow fashion. And in this project, we asked what would happen if we were to take all of these different types of measurements, really everything that we as a community know how to profile, and measure them on the same set of people. And we believe that if we did that, then because clinical symptoms of disease typically happen much after we would see some signature of them on the molecular data, then by profiling all of the types of data on the same people and following them for years, we'll be able to predict disease ahead of time. We'll be able to find new markers that appear before the disease and potentially be able to intervene in them and then develop new therapies. And of course, we'll be measuring things about people, so this will be very, very personalized. And ideally, this would lower the cost of drug development, which is very prohibitive these days. There's several billions of dollars that's the cost to bring a new drug to the market. And I think that with in a data-driven approach, we might be able to find new markers that are relevant for human, and hopefully also find new therapies in a much more efficient and less costly fashion. So all of this vision led to the idea of what we now call the Human Phenotype Project, like we had the Human Genome Project. So the Human Phenotype Project, which really aims to measure all different phenotypes that we can about people, leading to what we believe will be the next generation of precision medicine. And this is not just an idea, this is an actual project. that we've been working on for the past 5 years. We think of it as having 3 layers. The inner layer is the data, or we call it a library because library has the connotation of knowledge. On top of it, we build a data platform that I'll talk about later, which provides the access to the data. And this is what I wanted to share with you of how researchers here could get access and work on this data. And then once you get access, you can really look at many different discoveries and applications that you can build on top of it. So far in Israel, we profiled over 10,000 people. We follow them longitudinally, so we have over 35,000 human years that we've been following. So what is this data that we're collecting? What is this library that I'm talking about? So here you have all of the different measurements that we are doing. On the right-hand side, we have the physiological and clinical measurements, and these include historical blood tests that we take from people. So essentially, all of their electronic health records. We use ultrasound to look at fat in the liver. In the US, 30% of the adult population has fatty liver disease. We use the same ultrasound to look at the carotids for cardiovascular health. We use a DEXA machine that looks at full body composition and bone density. EKG and ABI look at cardiovascular health. We take high resolution images of the retina. So we have imaging data here, and I'll show you later that these high resolution images provide us with a view at blood vessels from which we can also look at various disease risks. We use sensors like continuous glucose monitors that for 2 weeks track the glucose levels of a person. While people are tracking their glucose levels, they also log all of their dietary information. So we have their very detailed dietary data. We use sensors, I'll show you later, to look at sleep. And we have several analog measurements, continuous measurements during the night. And I believe sleep quality hasn't ever been profiled on thousands of people at this resolution, coupled with all the other data that we have here. We have hundreds of questionnaires on medical background, family history of disease, lifestyle, and so on. And we have all of the past and current diagnoses of people. Alongside this clinical profile, we also have a very deep multi-omics profile, so molecular profile, and this includes a full shotgun sequencing of the human genetics, but then also shotgun sequencing of the microbiome, both the oral and the gut microbiome. We have metabolomics, so this looks at thousands of different molecules that circulate in our bloodstream. We do proteomics to look at protein levels in the blood, RNA sequencing to look at levels of RNA, and we have a novel immune assay that we developed where we can synthesize hundreds of thousands of antigens, and with sequencing, we can identify what the antibodies of a given person are recognizing out of those hundreds of thousands of antigens. So this, if you will, provides the entire infectious history of a person in just one experiment. And then we also biobank samples. We biobank blood and blood cells so that we can do future assays that don't even exist today. So this, as far as I know, is not the largest, but it's the deepest phenotyped cohort, namely the broadest number of tests that were ever done on such a group of people. We already completed in Israel profiling 10,000 healthy people in the age of 40 to 70, and also 2,000 people with various diseases where we collaborate with clinicians. And we have, we've been looking at 12 different disease areas like diabetes, oncology, various oncology cohorts, obesity, cardiovascular disease, and a few others. So this is the data that we have been measuring now. And I'll tell you, at the end of the talk, we have concrete plans to expand this internationally. One of the cohorts that we'd like to build is a cohort here in the UAE, and expand this into kind of a multinational, actually the first global multinational multi-omics cohort. So this data, as you saw, is very large, it's very diverse, it's very heterogeneous. And so in order to access it, we developed a platform. That platform is a secure platform where you can access the data, but you can't download the data, but you can do all the work that you want on it in a secure cloud environment. And that platform is going to be available in about 2 months' time. And that's the platform I'd like to share with researchers. We'd like to share with researchers here to for them to do and develop do work on this data. And I think a very interesting, very prominent case study for the interest that we expect to see from this data is the UK Biobank. Maybe people here heard about it. This has become now really the de facto standard of a healthcare data set, a very actually incredible project, 500,000 people from the UK, where this started about 15 years ago. They've been profiled longitudinally. So it's a very impressive data set. Tens of thousands of researchers are working on it, thousands of publications, not just people from the UK, from all over the world, people are working on it. But I think the data set that we have built and plan to expand is actually very complementary in that it goes much, much deeper in terms of the multi-omics profiling that we are doing. So I believe that there is going to be tremendous interest in accessing this data. If you think about all the different data layers that I mentioned, each of them on its own is perhaps the largest cohort for microbiome, for immune profiling, for RNA sequencing, but the power is in the combination of all of these different data types together. So what does this data platform look like? What will it look like? Basically, you'll get a research environment. This is an example of just a Python Jupyter notebook where you can see that you have here all of the different data types that are available, when they've been updated. And you can see, for example, here's imaging data from the retina. This is for just one participant. Here's the glucose profile of this participants with various events of dietary intake and meals that this person ate. So you'll basically get a research environment to explore and analyze the data. In the interest of time, I won't go into, of course, all of the details, but I'll just say that we have and are working very hard to do a lot of QC on this data and basically develop computational pipelines that take raw data like genetics data and eventually process it with all the various QCs. Each data type has its own QCs. So when you work with human genetics data, for example, you go from the FASTQ files into aligning them to the human genome and eventually determining variants, genetic variants that exist in each sample. So these detailed pipelines we have for every single data set. Here's a data set on sleep, for example. You can see this is, again, just one person for one night. We have 3 nights measured for each person. And you can see all of the analog signals that we have here for this person, respiratory events that he had during the night, when this person was awake, when this person was in light sleep mode, deep sleep mode, REM sleep, and so on and so forth. And there's also a very detailed QC pipeline that goes from the raw signal into processing the actual data that you will get in the end for research. Similar for the DEXA machine, which takes X-ray images of a person. We also have a pipeline. So this is really what we have for every single data set. So this is the data set that we have. And then my group has been working, obviously, on analyzing this data set to making various discoveries. I have in this talk multiple different projects that we've been working on. Obviously, each one of these projects is, sometimes a couple of students, sometimes more, working for several years to research one specific area. And what I thought I would do for the core of this talk is walk you through these various discoveries. There's multiple ones here. So what I thought I'll do today is, because we're limited in time, I'll just give a very high-level overview of each of these, just to give you a taste for the type of data that we have and for the insights that could be derived from it in various projects. And then tomorrow I'll be giving a workshop here, and we can go into much more detail and discuss the specific algorithms and even brainstorm about other things that could be done, or just in general answer questions about the data set and how people might work with it. And I'll just say, if you're students here who are interested in working directly on this data and having research projects, you can feel free to contact me also directly on that. So I'll go now through various projects. Again, they're going to be packed with a lot of information because I'm going through them very briefly. I don't expect you to, the goal is not to understand all the details, but again, just to kind of give an overview of the type of insights that we can derive from this. So one project that we did was on trying to take this largest microbiome data set that we compiled, a data set of 10,000 different people, several thousands of them already profiled with a second sample, and to try and develop a novel probiotics, so to identify novel bacteria that live among people that we believe could be therapeutic for various conditions. And so the computational pipeline basically starts from a discovery pipeline, which I'll mention in a moment, that tries to identify bacteria that may be causal for some diseases. And once we identify those bacteria, the idea is that because this cohort is actually a cohort that we built, we can go back to participants, get a sample from them, and isolate the relevant bacteria, and then eventually go into formulation of these bacteria and into clinical trials to see whether providing people with these bacteria could actually treat various conditions. The first two that we started working on are diabetes and weight loss. And the idea of this pipeline is similar to how one analyzes human genetic data. So with genome-wide association studies, where you try to search for associations between human genetic variants and host phenotypes. Here, we're trying to identify associations between genetic variants at the bacterial level and human phenotypes. So the illustration here shows just 3 samples where you can see that genetic variation at one position is associated with trait variation, in this case BMI, in these people. 3 people is just, of course, an illustration. We do this on the cohort of 10,000 people, and we find very significant results. This is a Manhattan plot like you see in GWAS studies, except this is now for bacteria are now on the x-axis, and the y-axis is the significance of the association. So highly significant associations, which plotted in a different way, show actually the size of the effect. So the number of BMI points that are explained by bacterial genetic variation. You can see some variations explaining 1 or 2 points of BMI. This can be even 10 kilograms of body weight. So it's a huge effect. To me, it's not surprising because we think of the microbiome as actually being an environmental factor, not a genetic factor. So environmental factors we know could very profoundly impact weight. But nevertheless, the strength of signals that we see here are much stronger than signals that we see in human genetics. And here's an example of one bacterial species for which we identified 2 different strains. Let's call them the blue and the green strain for simplicity. And we can see that there's almost a 2-point difference in BMI between individuals harboring the blue strain versus the green strain. And not only that, but when we look at what happens to these people 2 years later, we see that people with the blue strain associated with lower weight don't gain weight, whereas individuals that had the green strain, which may give, we believe, the propensity for weight gain, they indeed gain another point of BMI after 2 years. So the idea is to isolate now this blue strain and give it to overweight participants and see if this strain, in combination with several other similar strains that we're finding, can also give a benefit for weight loss. The same pipeline we can also use to try and find and discover novel antibiotics. This has become a major issue because of heavy use of existing antibiotics, has led to a lot of development of antibiotic resistance. And so there's a real need to find novel antibiotics. And so we can use the same pipeline to search for associations now between bacterial genetic variation and not human traits, but abundances and presence of other bacteria. And when we do that, we also find that genetic variation within a bacteria is typically positively associated with growth of that bacteria, because that we explain by bacteria typically evolve to grow better and faster. So we expect a positive association, but we also see typically negative associations between genetic variants in one bacteria and growth of other bacteria. And these presumably may be toxins or novel antibiotics that are produced in the warfare between bacteria, and their identification may lead to novel antibiotics, we hope. I mentioned that once we find these bacteria, we want to package them and use them to see if they can get clinical benefits. We've done this in one case, in the case of the disease of atopic dermatitis, a skin disease. And this is a paper we already published, a proof of principle study on just 9 patients. But in this study, we showed that participants who first receive placebo capsules with no fecal material, followed then by fecal material from a healthy donor, showed major clinical improvements in symptoms of the disease. This is one patient. Another patient, it may be hard to see on the slides here, but this data, as I mentioned, has been published. And when we quantify it, when our clinical collaborators quantify it, they see that there's essentially no change in clinical symptoms of disease after placebo, and a major, on average, 70% reduction in clinical symptoms once we give bacteria from a healthy donor to these patients. And when we look at what happens to the actual microbiome of people, we see that samples of donors are clustered somewhere in the space away from the square samples of the patients. And once we start to give these fecal material from the donors, we see a major shift in the microbiome of patients, and they become not identical, but quite similar to the microbiome of their donors. And this happens for almost every one of the patients. And so we believe that if we were to isolate these bacteria that got transferred, maybe this would be worth testing if such a formulation might actually be novel treatment for atopic dermatitis. So this is an example where we saw clinical benefits. Another example where we also studied the microbiome is in the case of chronic fatigue syndrome. So this is also a disease poorly characterized with not many, if any, biomarkers. And here I want to give you an example of a different type of data that we used on the score. And this is the data from the immune profile that I mentioned. I won't go into all of the experimental details here. They're also published in Nature Medicine last year. But basically, after synthesizing hundreds of thousands of antigens, we can take antibodies from the blood, from the serum of a person. And at the end, with next generation sequencing, we can say, what are all the antigens recognized by one's antibodies? And so when we do that, we find that actually people have, on average, they recognize about 1,000 different antigens. So their antibodies recognize 1,000 of these different antigens that we design. Some of these antigens seem to be specific to people, so only a small number of people identify them. But then there are some antigens for very common viruses, for example, that are actually recognized by a majority of the population, indicative probably of very widespread previous infection by these viruses in the studied population, in this case, the Israeli population. We also see that this immune profile is very stable over time. So different people have a very little correlation, again, mostly because of probably infection by common viruses, but the same people, even tracked 5 years apart, have an immune profile which is very correlated to the immune profile they had 5 years ago. So this is the type of data, and we applied it to a matched a cohort in chronic fatigue syndrome, matched, meaning that for every patient, we took a healthy person of the same age and gender. When we did that and applied the immune profile, we saw actually a group of antigens. In this case, antigens related to flagellin proteins. Flagellin proteins are proteins from bacteria that help the bacteria swim. They help them move. And so we think that what this result indicates is that in chronic fatigue syndrome patients, maybe there's more translocation of bacteria, which then produce some immune signature, and maybe that's at the very least a marker, and maybe even involved in the pathogenesis of disease. But in any case, we discovered here novel biomarkers for CFS, and could even develop a machine learning algorithm that quite accurately separates healthy people from diseased patients, and providing some diagnostic test for CFS that is based on specific bacteria that maybe could even be studied for therapeutic benefits. Okay, another area that we studied. So even though our main cohort is a healthy cohort, and even in a healthy cohort, many people take a lot of different medication. There's actually 2,700 participants that regularly, out of 10,000 people, take at least one medication. We have the information of all of the, on all the medication that people are taking, and we wanted to ask the question of whether taking the medication, which typically we know what the target of that medication is, it typically could be a human protein, but whether that medication might also have some side effects, because as we take it, it also is going to, whether we want it or not, it's going to encounter the trillions of different microbes that we have, and maybe it can alter their configuration. And indeed, we see that people that are taking medication, even when we match them by age and gender to healthy counterparts that are not taking that medication, we see that there's a major change in specific gut bacteria. So, for example, the diabetes medication of metformin shown here, there's certain bacteria that are really different between people who take the medication versus people who don't take the medication. So we believe that these are this is part of what happens when you take a medication, and this may in part explain side effects of medication, which we know exist on various at various levels. Because if you have a change in the bacteria, and we know that bacteria are very tightly involved in health and disease, then by affecting the bacteria, you might also have some effects on the host. So that's another area that we studied, and of course, medications could be linked not just to the microbiome, but to many different aspects of the data that we have here. Okay, I want to switch gears and talk about another type of data that we have. I mentioned it before. This is the high-resolution images of the retina that we take. So these would be examples of two, the left and the right eye of one participant. In this case, a 47-year-old male. And we have 15,000 images that we acquired, again, for 10,000 people, and for some measured in every 2 years when they came back for another physical test. And this high-resolution fundus imaging could really look at very look at blood vessels, could have a lot of information that exists in these images, and there's very exciting recent research into this type of data showing that you can look at micro vascular structures here, and that would be a window to the heart, a window to look at various cardiovascular health, and now we find also, I'll show you links to other phenotypes. So this is from a paper that was published just a few years ago, showing here some of these retina images for some people. And I don't know if you can see the details here, but I can tell you, for example, very accurate prediction of the age of a person, an error of just on average about 2 years. From these retina images, you can tell the gender of a person, you can tell whether he's been smoking or not, what his hemoglobin A1C levels are, so whether he's diabetic or not, what his BMI is. You can look at various blood pressure markers. So all of this you can understand just by taking a image of the retina. And it's also been shown in multiple other publications that from this you can also identify risk factors for various diseases. As you probably know, in the image space, now the state-of-the-art methods are deep neural network methods. And so when trained on this type of data, this is, for example, how the computer sees the image and can break it down into structures, into blood vessel structures. And so we built this type of pipeline that goes from the raw images, applies deep learning methods to eventually extract various features like the curvature, like the density, like the fractal dimension of these different images. And here's an example of, here's an example of images on the left-hand side from an older person, on the right-hand side from a younger person. And again, I don't know from this resolution, but you can see that even visually, it's very clear that as you age, on average, you actually lose or there's changes in the blood vessels. They become typically much less dense, and they change the structure such that you can really identify with quite good accuracy the age of a person. So these are, for example, some of the automatically extracted features, how they actually change with age, and you can see that there's various of these changes. When we took these features and asked whether they differ between people with cardiovascular disease or with obesity indicators, we actually found that various of these features are actually very different between people who have known and diagnosed cardiovascular disease. This is on the right-hand side, and people who have various markers of obesity, which we profile very, very deeply with, for example, the DEXA scans that we have in the data. We also identified, and this is novel because nobody ever coupled these retina images to continuous glucose monitors and to sleep monitors. We also identified that sleep quality is also associated with various features in the retina scans, as are various features on the continuous glucose monitors, on the basically the tracking of glucose levels in people. So I think this type of imaging data is very interesting. It's a relatively new type of data that's now becoming available, that people are finding has links to health, and coupling it now to the very deep phenotype data that we have in this cohort, I think provides us with many opportunities for new discoveries. Okay, another application, another type of data we're looking at are thousands of different small molecules, metabolites that we can measure by separating them according to mass with a machine called a mass spectrometry. And obviously, we know that molecules that circulate in our bloodstream have a major impact on our health, right? They travel systemically in the human body. So we'd like to understand what actually determines levels of different of these molecules. And so we search for correlations between changes in levels of these metabolites that we measure in people and various molecular factors that or other factors that we measured. And when we did that, we found that the gut bacteria are actually associated with changes in many different metabolites. The diet is associated with changes in many metabolites, and in other cases, its metabolites are associated with genetic variants in people. So by analyzing this data, we can now begin to understand what determines levels of different metabolites in the bloodstream. And this is a very important first key step if we want to, at some point, modulate and intervene and change level of metabolites to alter the course of disease, because we know metabolites are very important. Then coupled with this information, we can also go and identify novel biomarkers for disease. I'll show you a few such examples. So from also a paper that we published last year, looking at people who underwent a heart attack, people with acute coronary syndrome disease. Here, we could take even just a single patient, and we could go into our healthy cohort and find, for that patient, we could find a matched cohort. So for example, for a 60-year-old male who had a heart attack, we go into our data set and we find 60-year-old males who are healthy and did not have a heart attack. And that those healthy people become the personalized reference cohort for the patient. And so from that cohort, we can build the distribution of what we expect should be the levels of each metabolite. So for a 60-year-old male who is healthy, for each metabolite, what's the distribution that we see in healthy people? And we can then see what the level of that metabolite is for our patient. And we can see how whether it falls within the norm or whether it's outside. And we can do that for all the different metabolites and build a metabolite deviation table for every one of our patients when he's compared to the personalized and right matched cohort specifically for him. Once we have these metabolite deviations, based on the previous analysis that I just showed you, we can say whether these metabolite deviations are driven mostly by metabolites that are changed by diet, or changed by genetics, or by the microbiome, or by other risk factors. And by that, we can say, okay, so this patient has metabolite deviations that mainly are driven by dietary changes. So maybe the diet of that person is what led to the heart attack. But for somebody else, we would see that the metabolite deviations are actually driven by genetics or by gut bacteria, indicating that maybe even the treatment for these people should be different, because now by this analysis we can see what's common to the metabolites that change in these people. In a different story we also published last year, we looked at patients with multiple sclerosis, and here we found that on two different layers of data, on the layer of the metabolites and on the layer of the gut bacteria that produce a precursor to this metabolite, we found lower levels in patients compared to healthy controls. And this is in a pathway that eventually produces indole propionate. And indole propionate is a molecule that is known to protect neurons from oxidative damage. And so presumably, if you have less of this pathway, maybe this provides less protection for MS patients, and maybe also involved in the pathogenesis of disease. And we get more confidence because we find this on not just the level of metabolites, but also on the level of the bacteria that produce one of the precursors to this pathway. So just to show you an example of how multiple data layers could be integrated together. Then in IBD, we took advantage also of the very large cohort that we had. So we had 100 IBD patients, but we could match them to 1,400 healthy counterparts in terms of age and gender. And by that, go into a very robust analysis that identified multiple microbiome biomarkers that are altered in IBD patients, and also lead to a machine learning classifier that almost with perfect accuracy can separate IBD patients from healthy controls. Similarly, we have, as I mentioned, quite an extensive program in oncology. So we have various oncology cohorts, and here too, by finding matched controls in our healthy cohort, are finding novel biomarkers. In this case, these are biomarkers for pancreatic cancer, for breast cancer, in the microbiome space. Okay, one other example and story I want to share with you is on biological aging. So as you know, as we age, there's many different changes and many different molecular changes that happen. And so one of the things that we did was to take our data on metabolomics, our data on thousands of different molecules, and just see whether we can predict the chronological age of a person only by looking at these thousands of molecules in the blood. And turns out that we can. You can see the predictions here are actually quite good. But you can see that they're not perfect, meaning that there are people who have some chronological age, but the model at least thinks that their chronological age is older. And so we asked whether people who have, according to the model, a higher chronological age than they really have, whether that's indicative of maybe they're aging faster, and if they're aging faster, maybe they also accumulate more disease and more health conditions. And so indeed, when we compared people that, according to the model, have accelerated versus decelerated aging in terms of metabolites, we could see that people with presumably accelerated aging have worse clinical conditions. Okay? They have higher BMI, higher triglycerides, higher hemoglobin A1C, higher blood pressure, and higher liver enzymes. And so presumably, some of these would be novel biomarkers of accelerated aging. But metabolomics is just one of the many different data sets that we have on this deeply phenotyped cohort. We have multiple other body systems. And so we actually asked whether we can build such biological clocks, not just on metabolomics, but on all of the other here 14 different types of data. And indeed, we saw that we could generate predictions of chronological age that are significant. They vary in how much variance they explain by different data types, but each data type actually has you can build a significant predictor from it. There's also interesting differences between males and females. Typically, prediction of chronological age can be done better on females compared to males. We also see that typically, if you age faster in one body system, typically you would be at higher risk for a disease related to that body system. For example, if our biological clock that we build here for insulin resistance is older, then typically these people would have a higher risk for diabetes. If according to the DEXA, your bones, the bone density is aging faster, you'll typically suffer from low bone mass disorders, and so on and so forth. So in other words, these biological clocks that we're developing, they're really showing some abnormalities, some higher accelerated aging in a specific body system, and typically when that happens to people would tend to be at higher risk for a disease related to that body system. So it could be used as perhaps a diagnostic, and maybe something we could also eventually intervene in. An interesting difference, I don't have much time to go into this, but between males and females, you can see that in males we have a typical linear decline with age, but in females around the age of 50, we see actually a sharp transition, and this is just around the menopause age. And we see that even when we take a matched cohort of females who have passed or haven't passed menopause, we see that we can classify that based on various different data sets. So in other words, their blood lipids, their renal function, their immune system exhibit some alterations following menopause, which could be captured in these molecular data. We also see, interestingly, when we try to classify males from females, we see that our ability to classify that declines with age, or in other words, it seems that sex differences diminish as chronological age becomes older, and this is kind of somewhat known, but definitely not for all of these different body systems. Let me skip this one. And the final thing I want to share with you is results that we obtained from doing dietary interventions. So a few years ago, we started doing work just on the continuous glucose monitors and the diet data and the microbiome data. And what we showed is that dietary responses in terms of glucose levels to meals are highly personalized. So if we look at 4 people here eating bread, for example, we have some people who eat bread and have no glucose response, whereas others have a very high response. We developed an algorithm that could predict these, such that when we take people who are pre-diabetic and have many jumps and spikes in glucose levels, we could actually tailor a diet specific to them that would balance their blood sugar levels. And so we did recently a intervention where we took 200 people with pre-diabetes, we randomized them into either a diet based on our algorithm or a diet based on the standard of care, a Mediterranean diet. And we also collected many markers before and after this 6 months intervention. And we showed that typically people on the algorithm diet, they have glucose spikes shown here for 1 month in a pre-diabetic person that has many spikes that once converted into the diet based on the algorithm are virtually eliminated. And there's really clinical benefits that we showed here on primary outcomes related to spikes in glucose levels, related to hemoglobin A1C, and many other metabolic markers that improved. We saw that the changes that we did in the personalized diet, as would be expected, are actually much larger than the changes that one does in the Mediterranean diet, and many different food categories. I don't have time to go into that change, but one of the interesting findings that we also have is that we also see what could be construed as improvements in the microbiome of people, because we see that the richness and diversity of bacteria, and we also find specific bacteria that after half a year of intervention are altered in people who underwent the algorithm diet versus the standard of care diet. And we know that diversity and richness in the microbiome is probably a good feature. We also did some causal mediation analysis to show that some of the clinical benefits may actually be driven through changes in specific bacteria that may be interesting to extract in the future and potentially consider as specific therapies. We also are nearly completing a similar dietary intervention in women with breast cancer. This is an important problem to work on because over 75% of breast cancer patients are hormone receptor positive, so they receive endocrine therapy, but that therapy has a side effect of weight gain, which is bad in itself in terms of health, but also causes many of the women to stop in taking their medication. So it's important to treat this problem. We have nearly completed a randomized clinical trial on over 100 breast cancer patients. Again, the algorithm versus the Mediterranean diet. And not only are we not seeing weight gain, but we're actually seeing weight loss, not different between the groups, but in terms of the glucose levels, we are seeing major benefits for the woman with the algorithm-based diet. So this was, and I realize, a very packed, but I hope overview of many different discoveries that we have made, but that you can make with this type of data, hopefully to illustrate the potential. And I will say, this is just the tip of the iceberg, obviously, of what could be extracted from such a data set. And again, part of the motivation for me here today is to share with you this data and hopefully gauge your interest in working on it and accessing it in a few months' time when this is available and doing research on it. We are thinking that because of the major benefits that we saw from this cohort of 10,000 people, there's going to be a much bigger benefit for expanding this on multiple different axes. So expanding the number of people, expanding the different ethnicities and geographies in which we measure. So not just the Israeli population, but hopefully start a cohort here in the UAE, as well as in several other countries that we are discussing joining the Human Phenotype Project, but also profiling additional phenotypes. Even though this cohort is very deeply profiled, we don't profile everything yet. So there's additional tests that could be added. and of course, also additional diseases that we haven't been profiling so far. We developed everything in the group, from the recruitment of patients, the clinics, to profile them, the labs, to do the automation and high throughput and low cost multi-omics profiling, the biobanking, some with proprietary assays, and eventually the data platform that I mentioned, a secured cloud environment where one can study this data set and develop algorithms. And so we have all of this and are now looking to expand this and hopefully build what we believe will be the next generation of precision medicine. And I'll just put this acknowledgement slide of the many people in my group, research coordinators, lab technicians, data scientists who have been working on this project. And with that, I'll end, and thank you for listening.
Yeah, great part. So
thank you very much. Actually, we need to clap for this all work, for this amazing and great work.
Thank you.
So impressive. I'm so impressed. Your roadmap, you mentioned it's 40 years adults only, you following?
Right now it's 40 to 70
40
years.
to 70. Do you have plans for the future to actually start from birth?
So there's actually an interest. We're talking to the largest children's hospital in Israel right now. There's a strong interest in doing this on children as well.
Okay.
Yeah, I mean, I think there's so many directions one can go in. Yeah, absolutely.
Absolutely. I'm not sure whether you're aware, Dr. Ed, but G42 is part of the MRT Genome Project.
Yes, yeah.
So to date, we've recruited 300,000 participants in sequence.
Amazing.
So this would be absolutely, as I said, I'm head of biobank and director of biobank, so we're bringing on board proteomics as well next month.
Yeah.
January next year, I'll go live with that.
Yeah.
So everything you described, actually, we actually have under our umbrella. So would there be a partnership collaboration?
I would love to explore that. Yeah, absolutely. Yes.
Perfect.
Yeah.
Because if you're still okay, we will take you to.
Please.
And then I can introduce you to a few key people.
Please. Please. Yeah.
Yeah. Yes. Anybody who wants to come with me to work, welcome. Thank you, Dr. We're extremely impressive. I'm so overawed by your efforts.
Thank you.
Congratulations.
Thank you.
Maybe I can ask a question. So a lot of questions. Well, we know that the human body system have a lot of feedback. So in the clinic trial, using the bacteria from healthy people, right? How long actually will the trials? I was wondering whether say the bacteria will finally cured, right? In some way. So yeah.
Yeah, so I think when we go into clinical trials, one of the key questions is going to be whether the bacteria that we give actually colonize in people and for how long, and whether that's also person specific. So maybe in one ecosystem, one existing microbiome of a person, specific bacteria can or cannot colonize, but in somebody else they can. So I think we still don't know the answers to these questions, but I think now with this type of data, we can, for the first time, identify in a data-driven fashion bacteria that are relevant, and then go into trials. And then I think we'll learn how to answer all of these questions, which we don't have answers yet.
At least you know that bacteria are cohorts, right?
We believe we need to prove that in trials. But for example, the atopic dermatitis work shows that if you intervene in bacteria, there is a potential to also ameliorate some of the clinical symptoms of disease.
Thank you. Too long.
Okay, there's one question then online.
Yeah.
Dr. Shaker, can you go ahead with your question, please?
Well, first, I would like to thank Professor Sigal for this amazing talk, and thanks for all researcher talks for organizing such amazing talk. Actually, I'm Sheikha from the Biotechnology Center in Abu Dhabi, and we are looking to establish a multi-omic study. So hearing this talk with all this data is really amazing. My question was to understand whether you did the whole genomic sequencing or the whole exomic sequencing on it. And if we can have maybe another meeting with our biotechnology team where we can discuss further collaboration on such great project, and if we can help to design some of our model, especially that you have done a lot of work on the MS, and the team is also looking to conduct a special study for the MS patient here in UAE. Thank you very much.
Yeah. So thank you. So first of all, yes, would be happy, very happy to discuss a collaboration, which could be either, as I mentioned, providing access initially to our data set, which already exists, but also discussing the possibility of using all of the capabilities that we developed to help and profile more multi-omics on your cohort, if you don't have done that already. So happy to discuss that. And as for the question on genetics, yes, we have done here full shotgun sequencing, so full genetic sequencing. But I will say that just because of funding at the moment, this was done at what's called shallow sequencing, so at 0.7x of coverage. So from that, we could extract with 99% accuracy genotyped arrays, but also get novel variants. But now, as prices are coming down, we're looking into expanding the coverage and going into a standard 30x coverage of the genome. But so far, we have done full genome, but only at 0.7x coverage.
Using a long read or a short read, if you don't mind me asking.
So this is currently this is standard short read sequencing.
Oh, short. Okay, good.
Yes.
Okay, thank you. I'm looking forward to collaborate on this. Yeah, we would love to initiate another meeting to discuss all this. Thank you. Thank you.
I would be very happy to. Yeah. Thank you.
Okay, then thank you everyone for arranging this. I'll be leaving this meeting since I have another commitment, but thank you all for hosting such a great talk. Thank you very much, and a pleasure meeting you all.
Thank you, Dr. Sheikha.
Bye-bye. Thank you.
Are there other questions in the room?
Please.
Thanks for the nice talk. So you mentioned that the health people are also taking medications. So what's
the health?
So you mentioned the health cohort are also taking medications.
Yes.
What is the criteria of defining the health cohort?
Yeah, that's a good question. So it's actually very challenging to define. How do you define a healthy person? So after we have a doctors committee, medical doctor committee that eventually they defined it by exclusion criteria. So active cancer, active IBD, MS, various diseases that if one has, then he's excluded from the cohort. So because of that, you have many people taking various medications for IBS, which is not considered a disease for exclusion. Taking people with prediabetes taking metformin, for example, which is still not diabetic, but it's treatment for preventing diabetes. So that's why we have many people with medications that are still considered, and they are considered healthy people. So that's how we currently defined it. But we also have disease cohorts where obviously these people are taking much more medications.
Thank you.
Please.
Can you explain a bit more about how do you collect data? Like it is from one hospital or different hospitals, or what are the hurdles that you make because of some regulations and they are like privacy informations?
Yes. So the core cohort of the 10,000 healthy people, these people come to our study. They come to the institute and they sign an informed consent that for all the data that we collect, and that it can be used and shared and so on. So we meet these people and they sign an informed consent, and they upload their medical records. So this is how we work. We don't take existing data from systems, but it's like a research study where people sign an informed consent. It's also because the existing data is not well and good enough and not deep enough. It's not just the ethical issues which do exist, but it's also that we want to collect this very deep profile that doesn't exist.
In terms of giving access to the data, you
Yes.
went to the safe havens, that's
Yes.
model
Yeah.
biobank, because I worked with them myself.
Yeah.
I was part of the 100,000 genome projects as well, so
Mhm.
that's the one
Yeah.
that everybody's using.
Yes.
What hurdles have you had to overcome in Israel to be able to give access to that data to people outside?
So I think once we have an informed consent of
Yes.
people that they allow for that,
Sure.
then we can do it. But of course, we do it in a way that's very secure.
Mhm.
So under a secured cloud environment where you just technically do not have the ability to download the data, but you can do all the research into the data.
So access is personal.
Access is per yes, researcher or research organization.
I have maybe a naive question about that. So you have tens of the people data. So do you think that statistically the tens of the people data can cover maybe the major information of the whole human, or that the tens of data is not enough at all because there may be some data bias? And my second question may be that I do not understand more that's precious medicine. That what's your ultimate expectation of the more pressures? Is that means that accurate that for one people you can assign someone that's what agent that can diagnose or can do everything that a doctor can do, or what's the medicine that you design a medicine for one individual? That's my question.
Okay, so first of all, on the size of the 10,000, I think, well, I mean, as you saw in the results I showed, 10,000 is enough for making many discoveries, but I think there are some discoveries that obviously can't be made with them. So if you look at rare diseases, you know, that are 1 in 10,000 or 1 in even less, or even 1 in 1,000, you'd have 0 to just a few individuals and you can't study them. But I think on very common morbidities, like many metabolic diseases and cardiovascular diseases, when you have 10,000 people, you already will have hundreds and sometimes even thousands of people with that condition, and then that's enough to study them. But I think we want to expand the data set to a larger cohort, because then even for the common morbidities, we'll be able to look at effects that are smaller, which we can't do today. So it's I don't think that 10,000 is sufficient. I think it's good for some things and not for others. As for the expectation, if I understood the question right, for developing medicine, whether it's going to be personalized or not, I think ideally, of course, we're not going to develop a different medicine for each individual, right? But I hope that we'll be able to categorize people into, you know, maybe very broad 2, 3, 4, or maybe a bit more categories of people who really are different in the sense that the reason and cause for the disease is very different, and that means that the treatment really needs to be different. And I think the evidence today is that we have to have that approach, because we see that even all the amazing new cancer therapies are only working for 20-30% of the patients. So what about the other 70-80%? Obviously, we need something else for them. So ideally, we will be smarter as we collect more data, but ideally, there won't be a need to develop something tailored for each individual, but just to set groups of individuals with different characteristics.
Do you have how many pairs of twins do you have in the 10,000? Are you following?
We have, but this was not designed to look at twins, so we would have the rate that exists in the population. I don't know the exact numbers, but it's not very high for that. We do have some families. Sometimes we have spouses living together. In the age of 40 to 70, not too many, father, mother, daughter, son relationships, but we have a few of those. But we do have some households, which I think would be interesting to look at, maybe shared environment versus other effects.
Have you used the genetic reference map of Israel versus other
Yeah, of course. Yeah.
And what are you finding? Are there genetic differences?
Of course, yeah.
Yes.
You can, Yes, the Jewish population is distinct from
Absolutely.
Africans, Europeans, and yes, and even there you can separate easily between Jewish populations that came from different origins, the Ashkenazi Jews or the
Okay.
Sephardi Jews coming from different parts of the world.
Right.
Yeah.
Others? Yes, maybe it's not from the, I don't have any biology background. So
Yeah.
this is one thing about that, because that doctors are trained for 30 years just for one specific area, and you can collect all the information. So when you do the, for example, experiments or the analysis, you combine all the factors, maybe millions factors to analysis, or only for the factors that already in this field. Because I think that personalized, I do not have the like, human is a big system that every part are influencing together. But when people are doing something like a specific, they just analyze maybe one field or
Right.
some big pictures.
Mhm.
So do you have any experiment that can combine all things together and to see that how they influence each other, or that if something changes? And also another question is that you see the causal analysis. I do not think that when you have so many factors, you can do the full causal, because that's so many hidden variables
Yes.
to not discover. So how do you do the experiment? Do you have that the real the foundations that you can do the randomized experiments, or that how can you solve it?
That like a randomized experiment?
Yes, yes.
Yeah, so I think I showed in the end we did do some of that, like in the case of the dietary data. But I think you have to be practical and understand that you won't be able to do a randomized experiment for every causal relationship you want to study. So it's much easier to use observational data. Of course, you can't really infer causality, but you can make suggestions, and you know, that I think is how we should, just from a practical standpoint, how we should work.
Okay.
All right. In that case, so thank you very much once more.
Thank you.
Everybody rush off, they have lectures, that's
Sure, I'm sure. Yeah. Okay.